Hi, my name is Jane Wilkinson, and I'm the Senior Director in the Broad Genomics program, which is in the Broad Institute at Harvard and MIT. Biomarker discovery has really been everything about my career. Biomarker discovery created my career, especially in the genomics field. I started out as a team leader on the Human Genome Project. I was based at the Sanger Centre in the UK, and I was actually heading up the Chromosome 1 initiative for the Human Genome Project. It was a really, really exciting time and really kind of the first wave of genomics. Plant biomarkers actually called me across the ocean, across the Atlantic, and I started looking at cDNA function in crops, and that was working with Monsanto Company. And then I took a jump back to see out the end of the Human Genome Project at the Whitehead Institute, which is obviously now the Broad Institute. And now into this amazing world that we currently are in, where we can look at biomarkers across all different types of diseases and all different types of indications and traits. Genomics is really just an amazingly powerful tool for biomarker discovery. And, you know, I'm going to just kind of touch on a few examples of biomarker uses in disease treatment and trait discovery. So as an example, you know, we can now look at somatic variations in circulating tumor cells. Circulating tumor cells is almost like a liquid biopsy. Instead of having to do an invasive procedure to look at the cells that are in a tumor, or even to discover the presence of a tumor in the body, we can look at these circulating cells that are shed from the tumors, and that will tell us about the presence of cancer in the body, or also the progression of cancer in the body. It's really an amazing tool that's been recently developed and is really being used in the clinics now. We can also look at large-scale populations and search for clues around some of the very complex diseases. And a really good example of this in the neuropsychiatric world is looking at schizophrenia. We have a fantastic schizophrenia study at the Broad. And to give you a sense of scale and the power of biomarker discovery, our initial study was looking at 3,000 cases and controls, which sounds like a lot, especially from somebody who started out looking at one human genome. And you know, those 3,000 cases and controls didn't give us any loci that could be indicated in the disease. So we took that up a notch. We took that to 10,000 cases and controls, and we discovered 5 loci that could be indicated in schizophrenia. And then, you know, we take it up another level. We took it to 25,000 cases of control, and we've uncovered about 62 potential loci for the disease. So the power and the scale that genomics can give you in biomarker discovery is really, really important. And then to bring us into the current world that we're in, the COVID pandemic is another important example of the power of genomic biomarkers. We've got several studies underway looking at COVID positive patients and how their genomes may be impacting the outcome of the infection, or could also be impacting the type of medication and care that we offer those patients. A good example of interrogating a large data set is a project called the UK Biobank. They have hundreds of thousands of people already sequenced, and all they had to do was go and look at who was positive in that cohort because the genome data was already there. It's a really, really great study. And at the Broad and in Cambridge and Boston, we're also looking at some really important studies, especially looking at minority groups and also groups that are actually in different geographic locations, comparing their human genome and also their COVID viral genome and the presence of antibodies, pulling all of that data together to really kind of understand and characterize what biomarkers might be involved in also the progression of COVID in these patients. So I think I've given, you know, a good bunch of examples of how powerful biomarker discovery is, and also why, you know, even after 25 years of being in the field of genomics, it still very much captivates and thrills me. Thank you, everybody.